Welcome to the Bioinformatics CRO podcast. I'm your host, Grant Belgard. Today I'm joined by Dr. Adam Woolfe, a computational biologist and bioinformatics R&D leader at Bio-Rad. Adam's career has taken him from comparative genomics and gene regulation to single-cell technologies, antibody discovery, and computational immunology. His recent work includes Paraplume, a sequence-based approach to predicting antibody binding regions using protein language models. We'll discuss what he's working on today, the path that brought him there, and what he has learned about building a career at the intersection of biology and computation. Adam, welcome to the podcast.
Nice to see you again, Grant. Thanks for inviting me here.
So for listeners meeting you for the first time, how would you describe your current role and the questions you're responsible for?
Okay, so my role is basically to develop the bioinformatics tools, pipelines, and software necessary to develop our instruments and also to support the customers that use our instruments. So I think your listeners will be familiar with Bio-Rad. I think it's one of the biggest sellers of scientific instruments, things like digital PCR thermocyclers, gel electrophoresis machines. If you have worked in a lab, and I guess a lot of the people who do bioinformatics have not, but the people who make your data will probably know about Bio-Rad because it is such a big company. So I'm based in Paris, France, or just outside Paris, France. And how I ended up here was basically that I used to work in a startup company called Saber Bio. So I'm now part of the Saber Bio project in Bio-Rad, but Saber Bio was a startup company a couple of years ago. And we actually got acquired by Bio-Rad a couple of years ago to bring in droplet microfluidic single cell science into the Bio-Rad portfolio. So that's what we were doing. We were developing a technology called droplet microfluidics, which is basically the thing that drives a lot of single cell science today. People may be familiar with 10x Chromium machine to do single cell science, although there are a number of other technologies. But theirs is based on droplet microfluidics, and basically you're creating picoliter, like super small droplets that you flow through channels in a chip. And what we do differently compared to other people is that we have expertise in droplet assays. So we actually assay the function of the cell in the droplet using techniques to understand what's going in the droplet, and then we can use droplet microfluidics to sort the droplet down channels depending on the function that they have. So we apply this, of course, with the machine that we're developing actually is applied to identifying therapeutic immunoglobulins, so antibodies and TCRs. And what we do is we can assay the cells that produce those things, the B cells and the T cells, for the function that we're interested in. So for instance, for the antibodies, we're interested in binding, whether they bind something or whether they have a particular function, they can internalize or they can activate a cell. And all that can be done inside that little droplet using fluorescence signals and laser lines. And we can sort those cells. We can enrich for a population of cells that are producing molecules that we're interested in. And this is applied obviously to therapeutics, right? So antibodies make up like one of the most successful range of therapeutics, biologics, right, that are on the market. The value of antibody biologics is something like $350 billion today that could raise to $1 trillion in 5 to 10 years. In fact, out of the 5 of the top 10 biggest selling drugs, 5 of them are antibody therapeutics. In fact, the top drug, which is an antibody called Keytruda, made by Merck, basically brings in $32 billion to Merck every single year. So these things are big business, right? So identifying antibody therapeutics is a big deal these days. And so that's what our instruments, they don't necessarily have to be applied to that. You can use single cell science to understand other things as well. We work with groups looking at autoimmunity or whatever. But basically, one of the biggest applications, obviously, value-wise, is to identify new antibody drugs.
What parts of your contributions at Bio-Rad are least obvious from your job title?
So suddenly in a big company, you have to sell yourself you sell your ideas and the things that you want to do. You have to sell it to the higher management or to non-scientists, even to the marketing team. In fact, the marketing team is one of the most important aspects in a big company. Like, what is going to bring you value? You can, as a scientist, you have to think, the most interesting things or the most novel things are going to be the things that will propel you forward. But actually in a big company where the bottom line is whether you can make money from something, right, is more important. So part of my job is to explain the value of a bioinformatics function or a bioinformatics tool that will facilitate both the analysis of data coming from the instruments that we're creating and bring value and money to the company.
Could you trace a representative project from biological question through data generation and analysis to a final decision?
As I mentioned, I spend some of my time on R&D to help people prioritize antibodies. Okay, so when someone uses an instrument, they may end up with several hundred candidates that bind their antigen of interest or whatever. But the next stage is, which of those should I take forward to the next stage of testing and validation, which is expensive, right? Developing drugs is expensive. If we can spend time before, if the person can prioritize computationally the candidates that they can take forward, they can save a lot of time and money later on further down the road, because antibodies, as I say, are expensive to develop, and they fail very often because they have to be produced as drugs, and that process is quite rigorous. So they have to be produced at high concentration, high pH, high temperature, and that process can cause antibodies to aggregate and to lose their function or whatever. And so even if in the initial stages your antibody of interest was a super good binder, it doesn't mean that it's going to make a good drug, right? It can fail in many different ways. And so computational prioritization is a really huge field that is actively worked on by many people in the world today because of how much money you can save downstream. And so what we wanted to do is to get a handle on the fundamental function of the antibody, as it were. Okay, so what is the fundamental function of the antibody? It is to bind something in a very specific way. It's called the affinity of the antibody. And the active components of that are literally the amino acids on the outside of the antibody that interact directly with the antigens, right? So those are the specific interactions between the antibody and its cognate antigen. Those are the active components. Now, there are other parts, obviously, of the antibody that are important for ensuring that those amino acids are in the correct position, that they fold correctly, and that the whole molecule is correct. But those active parts are really the thing that drive the affinity. So if we can identify those, that would be good. Now, there were a number of different approaches that were already out there in the field, but they often were their accuracy was not great. And they often relied on structure, right? So structure is fantastic. It has revolutionized the field. The ability to, in silico, fold up a protein and know what it looks like in 3 dimensions, of course, is an amazing thing, right? AlphaFold has just revolutionized the field. And there are specific approaches to folding antibodies, like what AlphaFold brought. But that comes with a lot of computational resources that require you to expend to get the result. And if you want to do that on a repertoire level. So an antibody repertoire is a group of antibodies which can be in the scale of hundreds or thousands or tens of thousands of antibodies that come from a specific immunization or immune event from an organism that you need to characterize, right? So if you have a very computationally expensive process, you can only do stuff on a one-by-one basis. And this is true in anything in bioinformatics. If something takes 20 minutes per molecule to run on a GPU, okay, that's not going to be very useful. It's going to be useful to characterize maybe 1 or 2 sequences. But if you need to look at this whole process in a kind of repertoire level, and you can gain insights when you do high throughput that you can't gain when you're looking at things on a single level. We wanted to have the ability to look at these specific residues that are so important and look at them in a very high throughput way. So we wanted to make sure that the process that the model and the tool that we created was quick and was accurate, and it was at least as accurate as what was already available in the field. So we said, okay, there are these new things called protein language models, which are foundational models, which basically encapsulate a lot of evolutionary and structural and functional information in them because they're trained on millions or not billions, in the case of antibody sequences, so they already understand the fundamentals of what it means to be an antibody, what it means to be a protein, before you fine-tune them on things and say, okay, now you understand about proteins, now you understand about antibodies. I want you to learn what it means to be a paratope, right? So now we trained those models on a relatively small number of sequences because the availability of good, high-quality data in the field of immunoinformatics is actually quite limited. So you'll come across this problem all the time. There are just not that much data available that you can train your model on. So using these protein language models is pretty useful in that you don't have to give it the information from scratch. It already knows the fundamentals already, and then you just have to tell it, okay, these are the things that look like this. So we used 3D structures of real antibody-antigen interactions to train the model and see whether we could get a good accuracy. And of course, the novelty was that we didn't just use one protein language model, because there are a whole bunch of different protein language models that are trained on different data sets. Some are just on proteins, some are just on antibodies, some are subset of antibodies, whatever. They all have different types of information encoded in them. And no one model is necessarily the one model that should be used to train this. So what we did was we just stacked a whole bunch of them together, the embeddings of all these across the sequences, and then saw whether that would improve the accuracy. And in fact, it did. And using embeddings is pretty quick. So we were now able not to run one sequence in a minute, but like hundreds of sequences or 1,000 sequences in a few minutes. So now we can cover the repertoire in a very rapid manner in a way that we just couldn't before. And now trends that are interesting in that field, like just understanding how do paratopes change when an antibody becomes exposed to an antigen, right, in the body, when it goes from a naive to an antigen-specific antibody. How does the paratope, which is the active component of that, how does it change? And now, once we created that model, we were able to answer those kind of questions.
What are the most consequential false positives and false negatives in antibody or immune cell discovery?
Okay, so if you are using computational means to predict particular functions, obviously the consequences can be great. As I mentioned, the advantages obviously are that you can prioritize things quickly and you can choose antibodies that will likely not fail when you turn them into drugs. Now, the consequences of a false positive are that you can predict something that looks good. It may look like on paper that it works very well, but actually when you put it in the lab and you express it and you whatever, it causes problems because the model just wasn't accurate enough. Now, the false negative, obviously, is that you can miss things that were good, right? There's rare events that look possibly like a false positive, but in fact, it's not a false positive at all. And you can miss out on those things that would have made a fantastic drug just because the models or the predictions told you that it was not going to be great. So yeah, we still have a long way to go. Predictions are not biblical, right? They're just informed guesses, right? That you just have to take with a bit of pinch of salt. And some people maybe take them maybe too far. But I think there is a certain level of skepticism, I think, at least in the experimental community of AI models, which I think is completely justified. But I think if you only have the ability to test a small number of sequences because your budget is very expensive to do that, then I think computational prioritization is still a pretty good way to go.
How do you approach computational workflow design when an assay or instrument is still changing?
So this is something that I've spent literally the last 12 years doing. I was originally in a company called Hi-Fi Bio, where we were also developing droplet microfluidics screening for antibody discovery. We were doing it for internal screening rather than trying to create a machine that was used by people. But you have to go through so many cycles of changing the approaches from scratch. So effectively, you have to just be flexible and modular in your approaches so that you can take out certain parts and just modify them in ways that will allow you to deal with the new approaches. I think just flexibility in your software design and your analysis approaches that you can switch in and out of stuff so that you don't have to reinvent the wheel, but you certainly don't have to spend a lot of time changing stuff.
What does product readiness demand that publication readiness does not?
So publication, you have to be right once or a few times. Your experiment has to be right and robust enough to get through review. But it doesn't necessarily have to be true in every context, right? So I think there was a study done a few years ago that showed that something like 75% of published experiments were not reproducible, right? So it just shows that the requirement for reproducibility is something that is not generally done, generally because it takes time and money and effort. And once people have the result, they just want to get out there and publish it as quickly as possible, right? So they don't think about reproducibility. So when you're working in a company, reproducibility, robustness, and the fact that a machine has to be taken and used by different people in different contexts, in different environments, and it has to work every single time is just a completely different ball game that you have to deal with. So yeah, the two worlds, I think, couldn't be much more different. And in fact, A lot of companies are based on academic research that was done that looked promising. And they're like, yeah, just click your fingers and all we have to do is just develop this into a machine or into an approach and it'll work first time. And boy, is that wrong. The ideas are great, but turning those proof of concept experiments that perhaps drove a biotech startup where it takes a lot more to actually make it into a functioning product because it's just not as easy as people think.
What evidence makes you trust or distrust a model enough to let it influence an experiment?
Yeah, this is a really hot topic, I think, in the whole AI community right now, at least in science. I think if your model generalizes outside of the training milieu, like if it works on something that it didn't really ever come across before, that's the kind of the golden answer to whether your model is worth doing. Like there is a lot of cases, I think, where data poisoning occurs. So people are not necessarily aware that the data that they're validating on can look very similar to the data that they're training on. So if your test data has things that have high sequence identity or they are of same function, they may come through in the test data. And of course, the model just says, oh, I've seen that before. That's easy. So it elevates the model's accuracy rates. It looks like the model is much more accurate than it actually is. And in fact, when you test the model on data that looks completely different to the model, it often fails quite badly. So yeah, basically, if a model works well on data it's never seen before, I think it's trustworthy. Otherwise, not so much.
What's the hardest handoff among experimental biology, microfluidics, instrumentation, software, and data science? Where do you?
Yeah, so I think the handover, like I work with engineers, I work with cell biologists, I work with molecular biologists who are working on the instruments to make them work. And the handover of the data is trying to understand like the biases or the problems that propagate silently from the instrument itself to the data that you're looking at. It's like you don't necessarily have hands-on understanding of the instrument. It's hard enough to understand bioinformatics, never mind engineering and whatever. So spending a little bit of time in the lab, for sure, it goes a long way to try to understand the ins and outs of your data, like why you're seeing what you are seeing, and trying to understand that better so that you can mitigate the effects of those very specific aspects that you find in your data that are associated with the instrument that was creating it or the cell or molecular biology approach that was used to create the data. I think that's the most important aspect to understand. Yeah.
What changes when a development stage platform becomes part of a larger organization and what should remain unchanged?
Okay, so I've been through this process a lot. I've been in startup companies with a lot of change. And then, as I say, our startup company was acquired 2 years ago. So going through that process, you understand that the big organizations just work very differently because their aims are very different. Like when you're in a startup company, it's effectively proof of concept, largely. You have to work fast. You have to be prepared to fail, and it's okay because the cycles of development are much faster, right? So you don't have to talk to someone, you don't have to sell it, you just have to say, okay, look, this is what happened. I think we should try this approach or improve this, whatever. And then the people go back in the lab and do it. And so those cycles go fast. When you join a big organization, first of all, the teams that you work with expand, right? So you're dealing with people in a different country. Now you're dealing with people who are saying, okay, we need to get this product out on the market in 2 years, and it needs to be like this and this. Things have to be more robust. Things have to be more reproducible. Things have to be it's just a very different way of working. Sometimes big organizations, especially if taken over, if they've acquired a small biotech company, it can destroy the company. Like making that transition, especially if the team worked really well in that kind of dynamic environment, and suddenly when they have to work in a very much more formalized way, it kills something. It kills something in the motivation. It kills something in the dynamics of the group. And the company realizes it, and they go, okay, actually, we want to go back to the way that you were before because we thought you worked really well. So sometimes big organizations will say, actually, don't worry too much about the varieties that we have, especially in technologies that they're not used to, that they haven't developed these kind of things before. they don't have templates, right? They don't have things to say, okay, this is the way that we did it before, and this is because your technology is like something completely new to our organization. So actually, you're going to just let you get on with it, and because it worked pretty well while you were a startup. Yeah, I think that generally is the conversation that's had in these in big companies.
Now, a question that
Yeah.
is much discussed in our field. How do you expect AI will be impacting our field in the years to come?
There is already the big sort of hot topic in antibody discovery and development is obviously de novo antibody generation. So now not using screening technologies or phage display or hybridoma or whatever it is that people used to use to find their target is now using computers to actually create antibodies de novo, right? So using generative techniques to create an antibody that's never been seen before, and then using 3D modeling and other such techniques to try to predict which ones are going to be successful. Because as you can imagine, the search space in antibodies is absolutely huge, like phenomenal search space. So actually producing something generatively that might actually bind is a huge challenge. And now there are a bunch of different companies today that claim to be able to do this pretty successfully. There's companies like Chai2 and Nabla and all sorts of other ones. Now they produced white papers that look pretty impressive, but no one has actually seen the data because I guess it's so sensitive. Like the field is so competitive that they don't want people to see exactly how they did what they did. I think basically, as far as from what I hear, they basically throw the kitchen sink at AI models with as much interaction data as they can so that the model can basically then predict interactions. But antibodies are slippery. They're much more challenging than protein-protein interactions just because they're the active components of an antibody, which are basically their complementary determining regions. These are loops that come out of the antibody that interact with the antigen are quite dynamic, right? So they move around a lot and they can change their shape like before and after binding. So they're much more difficult to predict than general protein-protein interactions. So it's still a very challenging field. And I think, but AI seems to be every week, every month, there's new advances in this field. And yeah, the golden goose is to be able to predict an antibody very quickly, just de novo.
Where did your interest in computational biology begin?
I grew up loving computers. I used to do a little bit of copying magazines, like code. This was like in the 1980s, right? I'm pretty old. So I really loved playing around with computers. But I come from a very scientific family. Both my parents are PhDs, right? So science was quite a strong driving force in my career. And I never really thought about computers as a career. I always thought there's science and then there's computers, and computers are for games or, I don't know, software, whatever, and science is science. And I didn't really ever think about the two coming together. And it was only really so I did a degree in molecular biology at Manchester University. And as part of that, I spent a year in a lab at Hadassah Medical School in Jerusalem. And I remember one day one of my colleagues coming in and he was working on some bacterial protein and trying to understand something about why it was working. I can't remember exactly what it was. But he said, look, I've threaded my protein onto a 3D structure and it looks like it seems to be this kind of structure. And I was like, wow, that really blew my mind. Wow, you can actually take sequences and computationally predict stuff. That was my first ever exposure to that. And at the end of my degree, there were some courses in bioinformatics, and it just happened to be that Manchester was one of the first places in Europe to offer a master's in bioinformatics. And this was back in 2000, right? So I was in the right place by the right time because they were offering a fully funded master's. You even got some money to help you for accommodation. It's unheard of now, right?
Yeah, that's nice in the UK.
So they were offering that, and I was like, wow, this is amazing. I can combine computers with biology and two passions, together. And that's just a fantastic opportunity. So I did that master's. And then that is funny how these cycles happen, because at that time, people were talking about bioinformatics being this new hot topic, right? And that people were being dragged off the course to get jobs, right? Companies were so desperate for bioinformaticians that they were just dragging people off the bioinformatics course before they even finished, giving them high-paying jobs. And it's like, wow, this is going to be amazing. And I spent a summer working in a British biotech, which was an old biotech company that was quite well known at the time. And I said, oh, I'll finish writing my thesis and then I'll look for a job after, because clearly I'll just find a job very simple. I don't have to worry about that. And I graduated 2 weeks after September 11th, and the economy collapsed. Bioinformatics companies at that stage, the promise of bioinformatics just hadn't been realized. There was a lot of hype. There was a hype cycle, and suddenly people were saying, and it just was a little bit too early, right? The tools were not there. The data was not there. It was just too early on, and people had high expectations of this new field called bioinformatics, and it wasn't fulfilling. And so a lot of these companies were folding, and the people who had experience in bioinformatics were suddenly out on the market. And I was like, oh, I can't compete with my limited experience in bioinformatics. So I was unemployed for a year. And what I kept hearing was, oh, people have got PhD. No, I was like, at that stage, I didn't really think about continuing academia. I'd already spent 4 years doing molecular biology, 1 year doing masters of bioinformatics, so 5 years already at university. I was like, enough, I want to do something else. And you have a vision of your life that it doesn't always work out the way you think. And actually, I decided to go back and do a PhD because that was pretty much my only choice. And I basically was in the right place at the right time. I'm super privileged that I ended up studying the human genome just after it had been completed. Like I was given the opportunity to study the human genome and apply new bioinformatics techniques, multiple alignment, whole genome multiple alignment, using BLAST, whatever, just looking at comparative genomics and finding super interesting things about the human genome that was completely uncharacterized, right? You stuck a pin and you find something interesting. So I was just in the right place at the right time, and I feel very privileged to have done that, yeah.
And how did single-cell biology and immunology enter your career? What made them compelling enough to stick with it?
Yeah, they actually came in at exactly the same time. I hated immunology at university. I found it so boring. There's this cell, and there's this cell, and there's this receptor, and no, CD4. And you're just like, oh, seriously, this is just so boring. And it was not on my radar at all. And as I was coming to the end of my second postdoc, I was at the Institut Curie in Paris. This is how I ended up in Paris. Okay, so this is how a lot of scientists end up in countries that they maybe never imagined that they would be in, or that's a very common experience of scientists. I think outside of the US scientists tend to stay in the US, but scientists outside the US kind of move around. So I was finishing my postdoc, and I was wondering whether to stay in academia, but I wasn't sure, and hadn't really thought about I hadn't thought about industry either. I was still pretty wedded to the idea of an academic career because I'd had really fantastic experiences in that. But this startup company was basically wanted to pair with our lab to look at single cell chromatin, and we were the experts in chromatin at that point. And so I went to meet with them, and we discussed whatever, and I gave them my opinion and what I thought about the data. And then after the meeting, the guys, we're looking for a bioinformatician to set up the bioinformatics of our company. Would you be interested? And I was like, yeah, that sounds amazing. So this company, Hi-Fi Bio, was melding microdroplet microfluidics, so single-cell science, and immunology. They're trying to find antibody discovery all at the same time. So it's melding those two worlds in a single technology, and it just... that idea that you could use a technology to learn many different things, you can apply it to many different things. Also to TCR discovery, so looking at what you can do with technology that can assay function at high speed, just was very compelling. And in fact, what we started working on later on when Hi-Fi Bio became a therapeutics company was the field of immuno-oncology, right? So this is what really bit me, like the ability of the immune system to actually fight cancer, you can use your own immune system to fight the cancer rather than using an external drug, right? And I remember a couple of years into that, my mother-in-law actually got metastatic melanoma. She was diagnosed with metastatic melanoma, and there was buildup of a tumor on her, I think it was her kidneys or liver, and it was pretty fast. And they put her on this PD-1 immuno-oncology treatment. And lucky enough, because actually only 30 to 40% of people who actually undergo this actually respond. But lucky enough, she responded and her tumor disappeared. And I was like, that is amazing. This is amazing. And that really seeing the effects of something that your technology and your company can do is really compelling.
What's a career decision that felt least obvious at the time but became especially consequential?
I think it's that transition from academia to startup company. As I say, like when you're in academia, you can really feel like, oh, this is the be all and end all. And I think from my experiences were that what was happening in academia was far more interesting and radical than what was going on in companies. Companies were, for me, it felt like a few years behind the forefronts of science that were being pushed by academic circles. And I think that really was true in the early 2000s, especially with the Human Genome Project and everything else. Really, academic science was really pushing the boundaries. And at some point, I think those advances were now being pushed by startup companies, right? Like really incredibly interesting science was being done by those companies. And as I say, I wasn't looking to transition into industry, but it just for me it just happened, and I was like, oh yeah, this seems pretty cool, and I made that transition. But it was only afterwards that I realized actually I can make a lot more impact in a world in which the things that I do are actually translated into real drugs, real effects on patients, rather than in academia where things are, you make advances in science, but someone ultimately will build on that to help the people that you help. So in both ways. But I think that transition was not so obvious at the time and now is much more obvious. And in fact, if I look at all the kind of big names that were in the academic field when I was in academia and now actually in companies. Most of them have transitioned out of academia because I think they probably can see the same thing as I did, that a lot of exciting stuff is being done in companies now rather than academia.
What should an early career bioinformatician learn deeply today, and what can safely be learned as needed?
Oh, I think I'm a bit too old to answer this question because I've been doing bioinformatics for too long. And I think what someone does today might not be, or what someone might think is important today is maybe not what I think is important. But especially with AI, before AI came along, you had to do all the hard work yourself to find the answer to something. You had to look at what tools were available, what databases were available, and now AI can answer that question for you. But whether AI gives you the right question is still something that bioinformaticians, like having the sense to understand what is plausible and what is incorrect, is really where the bioinformatics will come in the next few years, I think, because AI can give you a lot of plausible code or whatever, but actually, when you know what the real answer is and how the tools that it's using should really be used, you understand that sometimes it just chooses the default method or whatever, and that's not necessarily the best method. So understanding the tools of the field that you're in deeply, I think, is the most important part, because then you understand how that tool should be used, because using just a slightly different parameter on a tool can make the difference between having a result and not having a result. And if you didn't know the difference and really understand how the tool worked, you would never understand why you were failing or why there was not a result at the end, or the result was not as good as it should have been. So obviously the fundamentals in bioinformatics, Linux command line, Unix command line, Python, programming, knowing how a server generally works in terms of memory usage and processing and all that kind of stuff. And then obviously the biology is critical, understanding like how to interpret the data you get at the end, because that is the crux. If you don't really understand why your result looks a little bit too good to be true, then you're going to present that as the result, and ultimately you're going to give people the wrong answers. So yeah, the thing is, it's such a huge field. I think it must be if you need to learn things very quickly, it can be overwhelming to know where to start. And yeah, I feel for those entering the field right now, because obviously it was much simpler when I started. We had like BLAST and a few cluster W and whatever, and that was it. And so I've spent my career learning all that stuff and that I can build on. But if you start right now, it's just, wow, where do you start? It's huge.
How should a scientist decide among academia, a startup, or a larger company?
I think it's really your style. Academia is great for looking at something in a lot of depth, choosing interesting questions that don't necessarily have a financial reward or a price tag associated with them. And I don't think you have to choose one or the other. I think you can transition between. I think transitioning from academia to industry is very easy. Transitioning back to academia might be more difficult, as academia often relies on your name recognition and your publication record, and sometimes that's not the priority in a biotech company. That publishing is a nice to have, but it's definitely not a prerequisite of being in a biotech company. If you want to work on something that's super interesting but not at big depth, it has to be good enough. Right? It has to be good enough to get the answer that people need to move on to make the decisions they need to move on to the next development cycle. And if you're okay in working in an environment, a high-paced environment where you're not necessarily doing something that is fulfilling scientifically necessarily, but fulfilling technical and team-building way, I think you can be fulfilled scientifically also in biotech as well. But it's just a very different style of working.
What's one piece of advice you wish you had received near the beginning of your career?
Be humble. Okay, like we as bioinformaticians, we are at the end of the process, right? We take data often produced by people in a lab and we analyze it and we give the result back, right? And sometimes we don't always appreciate the amount of time and effort that went into producing that data in the lab. The experiment may have completely failed. You may get 0 reads mapping to your transcriptome or whatever, or there's something else, something problematic, and you can go back to the person and go, oh, that was rubbish, complete rubbish. And you don't really appreciate like how demoralizing it can be sometimes to be told that something is rubbish. And for us, it was, we just put it through some alignment and then it didn't work, right? And it's like, okay, yeah, it's garbage. But like, you don't understand that person took a lot of time and effort to do an experiment and it doesn't really help them. So communicating the result to someone is almost as important as the result itself. So ensure that you are humble and that you are empathetic and understanding, and be careful with your words. You can sometimes you can come across as being harsh and it can be problematic. So yeah, if I would say to my younger self, just be a bit easier on people.
Which unsolved question at the intersection of single-cell biology, immunology, and computation are you most eager to see answered?
Yes, the thing that we're working on right now is trying to come up with models that can predict to some degree the affinity of an antibody to its antigen. This is a super difficult problem, and many people have attempted to solve it because, as I say, antibodies are slippery things. Their interactions with their antigens can be very difficult to predict. And in fact, also the place that the antibody will bind to on the antigen is also a really difficult problem. Like predicting the paratopes is not super difficult problem, but predicting the epitopes, the amino acids on the antigen that the antibody interacts with, is very difficult. The success rates currently of models on that is probably something like 30%, maybe 50% at the best. So it's really it's not a solved problem at all. And it's in fact a really important thing because people want to know, okay, so I have this bunch of antibodies. Sometimes an antibody can bind an antigen, but it has no effect. You want to maybe block an active site or a receptor or something like this. And if it's binding the site of it, it may be a good binder, but it doesn't do what you're really wanting to do. So knowing where the antibody is binding would be an excellent problem to solve because it would help people to just choose the best antibodies that are relevant to them. Again, developability of antibodies, that's a really difficult problem. Recent benchmarking showed that models are still really bad at predicting developability, things like aggregation or thermostability or polyreactivity or anything like this, are still quite difficult to do. And a lot of that is to do with dearth of data. Like antibody data is not very accessible, mostly because antibodies are so valuable, right? They sit in silos in big companies and you don't get access to them. So publicly available data sets that you can train off are just not very available. And so models have to be based on very small numbers of observations, which if you're using different technologies to measure that, it can come up with a very different result that will then screw up the model. So all these things are very difficult problems right now that I think would be great to solve. And we are trying to solve them ourselves. Yeah.
What would you like listeners to remember from this conversation?
I would like listeners to remember that life doesn't always take the path you think it does. You may have a plan. You say, I'm going to be a PhD, and then I'm going to open up my own lab, or I'm going to join a company or whatever. And sometimes you're thrown a curveball and things like global economy or just things that you would never imagine happening change everything. Even AI, I think, is just throwing a little curveball for a lot of people, and those entry-level positions are no longer available. So don't always imagine that one door shuts, another door opens. Your career path will often take a very different path to the one that you imagine, and that's not always a bad thing. And it may not be something that you originally thought you wanted to do, but it can end up being something that you're like, thank God that happened. I'm so happy.
Adam, thank you so much for joining us.
My pleasure, Graham.
The Bioinformatics CRO provides computational biology services to academics, research institutions, biotechs, and pharmaceutical companies on a flexible hourly basis. Visit our website, bioinformaticscro.com, to learn more.